हेलो एवरीवन, वेलकम बैक टू बायो एंड इंक। सो आज हम सब पढ़ने जा रहे हैं बायोएनर्जेटिक्स एंड बेसिक ऑफ मेटाबॉलिज्म, जिसमें हम थर्मोडायनेमिक्स एज वेल एज इक्विलिब्रियम के बारे में डिस्कस करेंगे। हम शायद अच्छे क्वेश्चंस या CSIR के क्वेश्चंस देखेंगे। ये बहुत ही बेसिक है, जो हर सेल के फाउंडेशन और कॉन्सेप्चुअल समझने के लिए चाहिए होते हैं। तो ये टॉपिक काफ़ी ज़्यादा क्वेश्चंस में पूछे जाने में से एक है, स्पेसिफिकली गेट के लिए। गेट में से इस टॉपिक से ज़्यादातर 2 मार्क्स के 2 क्वेश्चंस आते हैं। बायोलॉजिकल सिस्टम्स, एनर्जी कैसे यूज़ होते हैं, ट्रांसफ़र होते हैं, मेटाबॉलिज्म क्या है, उसकी टाइप्स क्या है, एटीपी करेंसी है, एनर्जी करेंसी है, उसका बेसिक क्या है, थर्मोडायनेमिक्स इक्विलिब्रियम का पूरा कॉन्सेप्ट क्या है, क्लास 11-12 में भी जो केमिस्ट्री में पढ़ा जाता है वो भी एक बार देखेंगे। सो ग्रैब ऑन योर नोट्स, स्टे फोकस, सब्सक्राइब बायो इंक एंड लेट्स अनलॉक अ न्यू चैप्टर विद इट। सो लेट्स स्टार्ट। अब मोस्टली जो पढ़ेंगे या तो NCERT के थ्रू पढ़ेंगे या फिर हमारा जो फेमस लेनिंजर है इसमें से पढ़ेंगे। लेनिंजर काफ़ी ही फेमस बुक है। अगर आपने नहीं देखा है या सुना है इस बारे में तो ज़रूर एक बार ये बुक गो थ्रू करें। एक बार तो ये बुक ज़रूर खोलिएगा। लेनिंजर काफ़ी ही अच्छी बुक है और थोड़ा सा एडवांस है लेनिंजर और NCERT में, बट एक बार आप उसको पढ़ेंगे मे बी उतने अच्छे समझ नहीं आए, फिर दूसरे तीसरे बार में यू विल गेट द ग्रास्प ऑफ इट। लेट्स कंटिन्यू। मेटाबॉलिज्म। सो हम सब ने मेटाबॉलिज्म क्या थोड़ा थोड़ा पढ़ा है। लेट्स स्टडी थोड़ा जस्ट में। ये एक हाईली कोऑर्डिनेटेड सेल्युलर एक्टिविटीज़ है, जिसमें मोस्टली हम मल्टी एंजाइम सिस्टम का बात करते हैं। ये हम देखेंगे आगे जब हम क्वेश्चन सॉल्व करेंगे। तो व्हाट डू मल्टीपल एंजाइम सिस्टम एक्चुअली मीन। अब मोस्टली ये जो हम बात करें सेल्युलर एक्टिविटीज़, इस सेल्युलर एक्टिविटीज़ में जो मल्टी सिस्टम एंजाइम्स होता है इज इन ऑब्टेनिंग केमिकल एनर्जी। एवरी सेल रिक्वायर्स एनर्जी, मोस्टली इट्स इन अ फॉर्म ऑफ ATP, बट वहाँ से पहुँचने के लिए देयर आर लॉट ऑफ मल्टीपल मेटाबॉलिज्म दैट इज गोइंग ऑन। वी ऑल नो अबाउट ग्लाइकोलाइसिस एंड Krebs cycle. सो ये एक फंक्शन है इस मल्टी एंजाइम सिस्टम का। सेकेंड इज कन्वर्ट न्यूट्रिएंट्स इनटू molecules from cell's own characteristics. Now these can be precursors to macromolecules. ठीक है। हाँ, मैक्रोमोलिक्यूल जैसे अमीनो एसिड्स, प्रोटीन्स, कार्बोहाइड्रेट, फैटी एसिड्स और न्यूक्लिक एसिड। अगर बड़े मॉलिक्यूल्स बन रहे हैं तो छोटे से बड़े भी बन रहे हैं ना, जैसे कि प्रिकर्सर्स तो चाहिए ही। और ऑल्सो पॉलीमराइजिंग मोनोमेरिक precursors to macromolecules. Next, also we see a lot of synthesis and ये सब, but let's dive right into metabolism का basic things. तो metabolism में हम mostly पढ़ने जाते हैं central pathway and how it is divided between different forms of life. ये काफ़ी divided होता है. What I mean here is कि someone is autotroph. We all know what autotroph is. Can you think about it? These can use the carbon dioxide from atmosphere as their sole carbon source. ऐसे कुछ एग्जांपल्स भी होते हैं, जैसे सिंथेटिक बैक्टीरिया, देन ग्रीन algae एंड सर्टन प्लांट्स, स्पेसिफिकली वास्कुलर। मोस्टली हम जब छोटे में पढ़ते थे हम एक ऑटोट्रॉप पढ़ते थे और हम एक हेटेरोट्रॉप पढ़ते थे। Now heterotroph is those that cannot use atmospheric carbon dioxide as their energy source. Now इनका नाम हेटेरो इसीलिए है बिकॉज दे यूज कॉम्प्लेक्स कार्बन्स। such as glucose. Okay, now इसका examples है certain organisms that are multicellular. Those are multicellular animals. Animals is one of the example. Okay, now एक ही difference है, mostly हम पढ़ते हैं बच्चों में कि autotrophs, these are photosynthetic organisms. Now these can obtain energy from sunlight and make their own food. While जो heterotrophs होता है ये काफ़ी complex है. These are known for obtaining the energy from degradation of organic nutrients. Now these autotrophs and heterotrophs in environment are interdependent on each other. So without autotrophs we cannot get our food. Very basic example. So just like it is said that suppose autotrophs, these use the sunlight, and then convert and build and also use atmospheric carbon dioxide. These use carbon dioxide and they in turn make the organic biomolecules. Now these biomolecules are used by heterotrophs. These biomolecules are used by heterotrophs. Now let's talk about one of the generated product that we see here. We all know the oxygen that is produced from water in this process. It's very important. That is how we get most of our oxygen, but also there are some small plants that produce way more oxygen. Now these heterotrophs, let me make man. These heterotrophs, these use certain plants, certain autotrophs, and then they consume the oxygen. There are different organic products that they use of autotrophs. That means organic products of autotrophs which help them further derive their own nutrition and return carbon dioxide to atmosphere. Okay, this is basic about autotrophs, heterotrophs. We are not going to dive into great details about what autotrophs, heterotrophs for the energy entropy. We are just going to understand what metabolism is and then further take it to bioenergetics and thermodynamics. So let's proceed. Now suppose I'm talking about metabolism. Now if we look at it, the basic definition is sum of all chemical transformation taking place in cell or organisms. Now these occur in series of enzymes catalyze reactions. Okay, so now these reaction-based enzymes that are catalyzed are also known as metabolic pathway. Okay, each of these consecutive steps in metabolic pathway bring a specific and a small change, but they help transfer mostly either, let's put this in points, they either help transfer, remove or addition of certain atom or functional group. Okay, so metabolic pathway is nothing. The basic thing is we are using certain different consecutive steps in which we are either do a very small change in transfer, removal, addition of particular atom or functional group. Now the precursor is converted into product. Now let's call these precursors as reactants. These are converted into products through a series of metabolic intermediates. Now this is called as metabolites. The intermediates are called as metabolites. Now further moving on to catabolism. Can you comment what catabolism means in the comment box so that I know that you know something about it? Now catabolism is degradative phase. What do we see in this? This is one of the most usual type of metabolism. In this, we have organic nutrients that are converted into, let's write smaller, simpler end products. Now this means, suppose we have carbohydrates and we have fats or proteins. Now these are converted or broken down into simpler end products such as lactic acid or carbon dioxide or ammonia. These catabolic pathways release energy which is conserved in formation of ATP. They release energy which is conserved in formation of ATP. Not just ATP, there are also certain reduced electron carriers. Now you must remember these because these are mostly asked in certain exams. A lot of people always forget that these also help in producing a lot of energy in the body, in the cell, sorry. These are NADPH, NADH, FADH2. Then a certain amount of energy is also lost in heat. Okay. Now further moving on to anabolism. If you have understood that kata means degradative, so what would ana mean? In a way you can say it's biosynthesis. From small, simple precursors to building up larger and more complex products. Now these require input of energy. Okay, now what it means is, generally in a form of phosphoryl group, the potential transfer of ATP, reducing power, and the reducing power of NADH, NADPH and FADH2. We make the utilization of these, we use ATP and we build up larger and more complex products. Next, let's go dive into. It is said if anabolism is, suppose, let's start with, if catabolism is converging, that means a small a gives B, C, D. Then anabolism is diverging. And this B, C, D, they all lead up to one simpler complex product. Now that you know basics about metabolism, let's dive into bioenergetics and biochemical reaction. Now you can watch this video in maximum 1.5x, but I hope to understand better, you would rather stick to 1.25x, not more than that. and anybody who wants to appear for competitive exams, do fill the CSIR form. Further ahead, you will study about bioenergetics, thermodynamics, chemical logic, certain biological reactions, certain oxidation and reduction reactions. So if you want, you can take 5 minutes or you can continue. Before that, we all know respiration. Now, respiration is slow combustion of carbon and hydrogen, which is very similar in even if you're burning candles or any combustible bodies. Now, just like that, our bodies also consume food and burn calories to produce oxygen. We are really going to review mostly the laws of thermodynamics, quantitative relationships, delta G, delta S, and delta H. That means free energy, entropy, and enthalpy. That means delta G for free energy, delta S for entropy, and delta H for enthalpy. Delta here means change. Now this change can be seen while reaction to product is converted, and certain changes seen in this to this. Even it could be the release of heat, which is seen through this. So moving on, bioenergetic. What is bioenergetic? It's the quantitative study of energy transduction. Now, energy transduction means the transfer of energy from one form to another. Also, this is seen a lot in living cells and all the biological energy transformation. Energy transformation obeys law of thermodynamics. We'll see this part a little bit first with NCERT and then with Leninger, which is a standard book that we use to understand basics and the complex portions. Okay, let's dive into certain thermodynamic terms, जैसे कि सिस्टम, सराउंडिंग्स, वैसे कुछ वर्ड्स होते हैं जो यूज़ होते हैं, उनसे क्वेश्चंस भी आते हैं. So a system and a surrounding. Now, a system is referred to a part of universe. Let's say universe हम मोस्टली सेट्स में यूज़ करते हैं, जब हम सेट पढ़ते हैं कि दिस इज़ अ यूनिवर्स, मतलब एक कॉन्स्टेंट ऑब्जर्व कर रहे हैं हम सामने से, उसके बारे में हम डिस्कस कर रहे हैं कि एक सर्टेन सिस्टम है, ये है सपोज सिस्टम, उसके आसपास जो भी है, लेट्स यूज डिफरेंट कलर, उसके आसपास जो भी होता है हम उसे सराउंडिंग कंसिडर करते हैं. Okay, now surrounding is something that is the remaining part of the universe, that doesn't get involved in system. एक यूनिवर्स सिस्टम प्लस सराउंडिंग से बनता है. Now, further समझते, suppose we have two substances A and B and we have kept these in a beaker. एक बीकर है जिसमें हमने A रखा है, एक बीकर है जिसमें हमने B रखा है. Okay, now they are both, these beaker contained reaction mixture and they are kept in a room. ऐसा हो सकता है कि एक these systems are kept separate by a boundary. Now, when we study boundary, let's make this as a boundary. This is a boundary. The red color is our boundary which separate system and surrounding. Okay. Now there are two types of system. One is open, one is closed. What is open systems? Now these have exchange of energy, exchange of matter very easily between system and surrounding. while the closed energy, they have no exchange. काफ़ी बेसिक है. Open means काफ़ी plug flow type का. Open means there are change, there are all the exchange in and out, and while the closed has a tight grip on its boundary. Okay, there are also a third type of system, that is isolated system. Now these are there's no exchange happening of matter or energy, but also the presence is of such things are highly contained. One of the example is thermoflask. Yeah, properly insulated and closed vessels. These are the examples of isolated systems. तो नेक्स्ट कुछ क्वांटिटीज़ पढ़ते हैं. Let's study internal energy. Now this is denoted by a U. Internal energy. Now इसमें what happens is we consider the changes in heat, then work done, then matter. Okay, so this is a quantity that represents the total energy. That's denoted by E. Total energy of the system. Now the heat passes in or out of the system, or work can be done on or by the system, or matter enters or leaves the system. These are certain conditions that we see while we are studying total energy, which is referred to as internal energy. Okay, when we study about heat, we know that heat, when contained in a thermos flask, suppose I have a thermos flask. Now in this there is no exchange of heat. So what we see here is no exchange of heat within the boundary to any system or surrounding. Here we see a term that is called as adiabatic system, which has no exchange of energy from its boundary. That means suppose we denote heat with Q is equals to 0. There is no exchange of heat. Suppose I have a container and this is the surrounding. Now there is mass and matter change, but there is no change in heat. Q is equals to 0. Okay, now this is surrounding, this is system. Now let's clearly state U is internal energy and T is temperature of a system. Okay, so if there is a system or there is a state of system and that is A, so we note it by UA TA. Okay, suppose for a reactant UA is the initial stage and UB is the final stage, as in the product is changed from A to B. Now we have changes in internal energy. We calculate it using delta U, which is equal to UB minus UA. Always remember that the changes in the internal energy of a system after a reaction would be more, because we see that during a reaction there are changes in chemical bonds, then changes in bond length that affect the internal energy of UB. Okay. Similarly, if we keep the second parameter, temperature, now when we notice that a substance changes from A to B, the temperature sometimes could be high, could be low. So let's put a condition where temperature of B is more than temperature of A, then the change of temperature that is calculated is using TB minus TA. These are some basic way we structure a reaction. I did explain you about when work is done on, which means then work is done on a system. In the system you maybe are using a mechanical stirrer or anything, so you are doing work on this system, so we use a positive sign for work. Now when work is done by a system, suppose we use a sign negative. When work is done by a system, we use a negative sign because we see that there is a decrease in internal energy. Okay. Now let's discuss about heat. Now the heat is exchange of energy with a result of temperature difference. Change of energy with an exchange of heat is exchange of energy with change in temperature. Okay, that means let's denote heat with small q. We see that initial stages and final stages of a certain system can be different, considering if there is exothermic or endothermic reactions. Now let's see one more term, that is enthalpy. For enthalpy, this is one of the thermodynamic terms. We use this as let's write a basic formula for it that will help you understand what enthalpy is. H is equals to U plus PV. Now this is seen to understand that how enthalpy is regulated with internal systems as well as volume and pressure of the system. It is important to understand that heat is absorbed by the system when there is or at a constant pressure, and that is how we measure enthalpy. Also remember that enthalpy, that is delta H, certain changes in enthalpy is denoted by delta H. Enthalpy is negative when we have exothermic reaction, and delta H is positive when we have endothermic. Always remember enthalpy TH and exothermic TH. These are used to understand how the energy evolve or absorb from the system. Also we see heat capacity. Which is measured by Q is equals to C delta T. Now C is heat capacity of a certain system or a certain substance. Suppose we have a system at a constant volume, then QV is equal to CV delta T, which is equal to delta U, that is internal energy, and we have constant pressure. Then we have QP, which is equal to CP delta T, which is equal to delta H, enthalpy. Now we know that measurement of delta H and delta T is seen in calorimetry. We have all done that measurement in our labs. So moving on, just a revision. In an exothermic there will be negative sign of delta H, and in endothermic there will be a positive sign. Similarly, that happens for QP. QP is positive for endothermic, which means when delta H is positive, and QP is negative when there is exothermic or delta H is negative. Okay. Now one more term, that is enthalpy. that is a reaction when converted from reactant to product, they have certain enthalpy change, which is known as reaction enthalpy. Now this can be summed up with a symbol delta HR, which is denoted by sum of enthalpies of product minus delta HP. Let's denote delta HP. Let's show the change from first enthalpy reaction by delta H is equal to summation of sum of the products of enthalpy of products, sum of the enthalpy of products, that is delta HP minus delta HR, which is sum of enthalpies of reactant. Now we use sigma to summate all the reactions for a product and a reactant in a stoichiometric reaction. This is how I would explain it with basic words, basic terms. Now moving on to Leninger, this is how it's written in NCERT and how it's taught in class 11. So if I have to speak you from Leninger, it will be short and graphs like a mind map. So okay, stay tuned. So when we are studying it from Leninger, we're not going to study it one by one or one term, second term. We're going to accumulate and study. Like first start with first law of thermodynamics. Now that you understand basic terms of thermodynamics, let's dive in about the laws. Okay, so first law states that for any change, physical or chemical change, there is change in the total amount of energy in the universe remains constant. We just studied what a universe is, so I don't think it will be difficult for you to understand. The total amount of energy in universe is always remaining to be constant. Okay, in a way we are saying that if they remain constant, so it cannot be created nor can it be destroyed. Okay, let me revise it again. The first law of the principle of conservation of energy for any physical or chemical change is that the total amount of energy in the universe remains constant. The energy may change in its form. That means energy may change in form, but it may be transferred from one form to another, but it cannot be created nor destroyed. Can be transferred, but it cannot be changed, or neither can it be created or nor destroyed. Now the second law of thermodynamics. Now this states that the universe tends towards increasing disorder. Now when this word increasing disorder is connected, we see a word that is related to thermodynamic in thermodynamic term, entropy. Now this is denoted by S. So this is a quantitative expression of randomness or disorder in a system. Okay, let me again repeat it. So the universe always tends towards increasing disorder. In all natural process, the entropy always keeps remaining. The entropy always increases in a universe, always increasing, plus S. Okay. To understand this law, we use three thermodynamic quantities. That are Gibbs free energy, which is denoted by a G, then enthalpy, which is, yes, it's denoted by an H, and then entropy, which is symbolized by S. I want the video to be more lengthy, so subscribe to Bio and Ink and stay tuned for such more videos. And after we finish one topic, we would like to go with certain specifically because CSIR is coming, so we like to do PYQ that is related to CSIR. So stay tuned and follow and subscribe, and do comment if you like the video, and if you want to keep continuing, do let me know if you like the content. Thank you.